Hello, I'd like to talk to you about schistosomes, which are intravascular parasitic flatworms, and in particular how they deal with host pruinergic signaling molecules like ATP. In the extracellular environment, ATP can act as a DAMP, a damage-associated molecular pattern released by stressed and damaged cells, and it is pro-inflammatory. The worms have enzymes on their surface that can cleave ATP, and one of them is ATPase1, cleaving ATP here, and also cleaving ADP here to generate AMP. The worms possess a second exposed ectonucleotidase called NPP5 that can also cleave ATP here and ADP here to further generate AMP. Recall that ADP is prothrombotic, activating platelets, so the ability of these enzymes to cleave these host signaling molecules we believe would benefit the worms in the vasculature. A third exposed ectoenzyme is alkaline phosphatase that can remove the phosphate from AMP to generate adenosine, which is anti-inflammatory. Again, we imagine this benefits the worms in situ. Another pruinergic signaling molecule that the worms may encounter is NAD. It can be immunomodulatory, and this exposed enzyme NPP5 can cleave NAD to generate these products NMN and AMP. The worms have a second NAD cleaving enzyme called NACE that can remove the nicotinamide moiety from NAD and also generate ADPR. And these products can be further acted upon by these enzymes to generate, again, nicotinamide ribose phosphate, and here generating AMP and ribose phosphate. Now we've established that alkaline phosphatase can act on AMP to cleave the phosphate and generating adenosine, which is a common reaction product of these pathways. So in sum then, we see that this series of ectoenzymes on schistosomes has two main functions. It can get rid of these powerful signaling molecules that the host produces, and also it can generate useful molecules for the worms. We've talked about adenosine, but in addition to it acting as a signaling molecule, we remember that schistosomes, like many parasites, cannot synthesize purines de novo, so they must take up molecules like adenosine in order to survive. Likewise, the nicotinamide that's generated is a form of vitamin B3 and is essential for the worms to survive. So in sum then, we have a series of ectoenzymes that are controlling this collection of pruinergic signaling molecules around the parasites. In other words, they're controlling the schistosome's pruinergic halo.